A warm welcome to all. So welcome back again to the class of cytology. So today let us discuss our next cell organelle, that is mitochondria. Okay. So now you are familiar with the cell wall, plasma membrane and some of the organelles like endoplasmic reticulum, Golgi complex, etc. Okay. Now let us discuss another very important cell organelle that is present both in the eukaryotes, that is like animal cells as well as in the plant cells. Okay. So this mitochondria is actually a small cell organelle and that is first observed by Kolliker in 1815, and the name mitochondria is given by Benda in 1897. Okay. And they are usually absent in prokaryotes and also in anaerobic eukaryotes, but present in plants and animals because both are eukaryotes. Clear? Then cells of the dormant seeds also having very few mitochondria. And mitochondrial size also may vary depending upon the species. Okay. So mitochondria in generally green plant cells contain less number while comparing to the non-green plant cells and animal cells. Okay. So green plant cells, the number is very less. While discussing the shape and size, you can see usually the mitochondria having a somewhat cylindrical outline and the size is also variable, generally in between 1 to 4.1 micrometer, and the diameter is having 0.2 to 1 micrometer. Okay. Now, what are the chemical constituent of mitochondria? The mitochondria contains proteins, lipids, RNA, DNA. As it contains its own DNA, that's very important point you have to discuss. Mitochondria have their own DNA. That's why this organelle is known as semi-autonomous organelle. So why mitochondria is called as a semi-autonomous organelle? Because mitochondria having its own DNA, so it can do all the activities that are done with the help of DNA. Clear? So that's very important point. And it also contains minerals, trace elements, magnesium, calcium, phosphate, etc. So while discussing the structure of a mitochondria, you can see, I will show you the structure. You can see there are these outlines somewhat, I already told you, somewhat cylindrical in structure, and membrane is having, there are two membranes, that is why outer and inner chambers, you can observe two chambers. This is the outer chamber and there is an inner chamber, and the inner chamber is having that foldings like this. See, foldings like this all over, and these membranes having a thickness of 60 to 75 angstrom. Clear? And in between the outer and inner membrane there is a space. You can see the space. This space is known as perimitochondrial space. Clear? Okay. So this is the outer membrane, and this is the inner membrane, and this is the perimitochondrial space. Clear? And inside this you can see lots of fluid and also the circular DNA associated, and fluid also present, that is known as matrix. Clear? Okay, let us discuss in detail. See, what is about the outer membrane? So outer membrane is very smooth, permeable to large number of metabolites. They can enter inside the mitochondria. And there are protein channels in the membrane, that is known as porins, or minute pores or openings or protein channels in the membrane. Then while discussing about the inner membrane is permeable to only some of the metabolites, not all, but only some of the metabolites. And in both these membranes usually rich in phospholipids. So outer membrane, inner membrane, outer membrane smooth, inner membrane and permeable to almost all metabolites. Inner membrane is also permeable to only some of the metabolites. Clear? Now see, in this image you can see how these are organized. See, this is the outer chamber and this is the inner chamber, and inside this inner membrane they are having foldings. The foldings of inner membrane is known as cristae. Clear? And cristae having some other structures all around it, that is known as oxysomes. So inner membrane is folded into various foldings known as cristae, and cristae is having, they are for increasing the metabolic activities and an active area of metabolism, because it's a widely spread area is increased, surface area is increased, and also cristae having a small tennis bracket like structures, that is elementary particles, or you can call it as F0 or F1 particles, or otherwise known as oxysomes. In the last picture you have seen that this is the oxysomes or F1 particle, and these are the cristae or the foldings, cristae having the oxysomes or F1 particles. Clear? Okay. Now the F1 particles having a head and a stalk. Clear? It having a head, that you already know that F1 particle, this is take an example, see like this, a head and a stalk. Okay. And that is base is the F0 subunit, this is the F1 subunit having 1 millimeter long thickness. Stalk is 5 nanometer long. Okay. And the head, that is F1, having a diameter of 8.5 nanometer. So that's a head and a stalk. Now about the perimitochondrial space. What is perimitochondrial space? It is a space between the outer membrane and the inner membrane. And the space is, it is a very important part, and it extends the space of the cristae, and it contains a semi-fluid. Inside this membrane there is a semi-fluid matrix, and matrix has protein particles also. Matrix contains the DNA, RNA, ribosomes, enzymes, etc. And these enzymes are very important for Krebs cycle. You know what is Krebs cycle or TCS cycle? Tricarboxylic acid cycle. Okay. And DNA makes this organelle a semi-autonomous, that I already discussed. So, two membrane structure, outer membrane, inner membrane. Inner membrane is folded inside, increases surface area of activity, and that is having some particles known as F0, F1 particle. And in between the membrane there is a space, perimitochondrial space, and inside the inner membrane the matrix that is filled with enzymes, DNA, RNA, and so on. Okay. See. Now mitochondria's DNA is very important, that is double stranded DNA, and it has its own ribosomes also. Mitochondria having its own ribosome also, and the mitochondrial proteins are synthesized under the instruction of nucleus. Okay. So they are synthesized under the nucleus, instruction of nucleus. You already know what is protein synthesis. It synthesizes their own structural proteins also. This is the ultrastructure. You can see cristae and particles attached on the cristae, outer membrane. This is a space, perimitochondrial space. This is the inner membrane, and the ribosomes present, DNA present, everything present inside the mitochondria. Okay, so this is structure of mitochondria. And mitochondria function, when discussing, they are considered as the, they are the ATP synthesis production factory or energy factory. You can call it as an energy factory. They are the miniature of biochemical factories. They are the powerhouse of the cell. You can call it as powerhouse of the cell. Okay. And most of the cellular respiration that part take place inside mitochondria. And energy liberated in the process is stored in the form of reduced coenzymes, ATP, NADP, etc. So they provide an important intermediates for synthesis of several biochemicals like chlorophyll. Okay. Then many of the amino acid synthesis occur inside this, and they are store and release calcium when required, and also factory of enzymes, and that synthesize fatty acids, so on. So that's a function of mitochondria. Okay. So I hope you understood well about mitochondria. In brief, mitochondria is known as powerhouse of the cell, energy factory, very important structure, outer membrane, inner membrane, in between membrane, the space, perimitochondrial space, inside inner membrane, the matrix filled with enzymes, DNA, RNA, and other factors required for energy metabolism, and also that inner membrane having foldings known as cristae. Cristae is having elementary particle F0, F1 particles, and they are the active site of ATP synthesis. Okay. So that's all about the cell organelle mitochondria. So have a nice day. Thank you.